Today, this shouldn't take the entire time, but what I really want to talk about today, in preparation for your final manuscript, is to talk just mostly today about writing mechanics, kind of stay away from content. If you do have content questions later, since only my class is here, we can do some content questions later, but I really want to focus the first part on writing mechanics, and that's why I brought Dr. Brannick, Dr. Santiago into the room with me, so they can kind of give their insights on writing mechanics. Okay? So for most of you, you've written only really one other big research paper before, correct? That was in Gen Bio 1. What was the topic of that research paper? Salt stress, right? So it was kind of amorphous, right? It was kind of like, what am I doing? Well, how am I doing it this way, right? Okay. How much instruction did you have for that paper? Did they just kind of go? Some of you probably had a decent amount of instruction.
I'm just curious of what my students said for how hard is that?
Okay.
Brandon.
So you wrote the salt stress paper. That was like the first big research paper that you did. So now this is Gen Bio 2, so we expect a little bit more out of you, right? You're in my class, so I expect even more out of you. All right, because my class.
No pressure.
There is pressure. We're not trying to. What are you doing, pressure? Okay. So the best way probably to learn about writing mechanics is to actually do it. And the first thing that you really need to know about scientific writing, rule 1, it should be the most boring thing that you've ever written in your life, right? It should be like, take out every flashy word, adjective, things that you normally would use in creative writing and make it really boring. The reader should be intrigued and bored 100% at the same time as they're reading it, because we try to say things in the least amount of words possible when you write science. So you want to say it once and say it well and not say it again. So that brings me to my first kind of key point of things that to look out for when you're writing. Redundancy is huge. If you said it once, you don't have to tell me again. For example, we did 2 different rounds of PCR in here. You don't need to tell me what PCR is twice. You should tell me that you did PCR twice, but you don't need to tell me what it is twice, right? So there's certain key aspects. I'll get your question. Yes. So there's certain key aspects of PCR that should always be said, like what are some examples of those sorts of things? Concentrations. What else should be said in a PCR? When you say I did PCR, what other things should be said in that description?
The kind of
the primers that you used, right? But you don't need to tell me, when you say PCR, you just say I did PCR. You don't need to tell me PCR involved these steps and all this other stuff, right? So that's understood, but then when you, like for example, in my paper, we did PCR twice. You don't need to say, go through all those things again. You can just say, have a section in your materials method say PCR, right? And tell me, like in one sentence, what you did for the first genotype of PCR, and another sentence, what you did for the second genotype. So we want to cut down on any sort of redundancy in there. Same thing with gels. We ran a lot of gels. You don't need to tell me every step-by-step instructions on how to run a gel. I know how to run a gel. So when science writing, when you say things like PCR, gels, common techniques, when you say I did that technique, there's things that are understood when you say I did that technique, like I did a gel. I'm going to go out on a limb and say, yeah, you ran a gel, and that gel had agarose in it, and that you looked at it under Lycor, and you saw a size of a band, and that told you something, right? You don't need to tell me that I measured out 1 gram of agarose and made this thing and did all of it. That's understood, right? When you say agarose gel electrophoresis. So keep that in mind to keep the words down, especially because most of the questions I'm getting so far in my office have been about materials and methods. It's like how to be succinct without leaving out important details. So when he goes to write the materials and methods section, make sure you say everything that you need to say in the least amount of words possible. And then that will be your best course of action. So a good way to write a materials and methods section is to write everything and then start cutting. Saying, is this important? Is this too wordy? Can I say this? Is the sentence 10 words and it can be said in 8? Does anybody know a reason why we do this in science? Why we're so careful how we choose our words and
more times that you might say something, you might alter something slightly, and that could create confusion.
Yeah, you might create confusion. You want to limit confusion. What's another reason? You have
Read and be done.
read and be done. Okay. Be done. Okay. What are you thinking, Dr. Santiago? Money drives everything, right? So a lot of journals have what they call page charges, or I'll fix that in a second, or they have word charges. So sometimes in a journal, you're limited to 3,000 words. So every single one of those words matters. So you don't want to be redundant in anything, because if, for example, you go over by words, they might reject your paper, or they might tell you to cut it, or they might tell you to put it in supplemental, or they might just, worst case scenario, reject it outright for not following instructions, right? And yes, this happens to us too, like how if you don't follow instructions, I may give you a 0 or give you a bad grade on something. In my world, if I don't follow instructions, if I don't get something in on time, by the deadline, or follow the instructions to the T, they throw it in the garbage. And they say, thanks for coming, please play again, right? So this is why you might be wondering why we care so much about this, is because we're trying to teach you to do it the right way. Okay? So the best way that I've learned how to teach you and give you examples of this is actually pull up a paper, and we're not going to read it for content, but what I like to do is I like us just to look at different things about the paper and see how they structure the writing style of the paper. So here's the materials and methods section of this particular paper, right, on the right-hand side here. Just take a second. You do not need to know what they did in this paper, but just look at this first paragraph for structure. So what do you see structurally wise in this first paragraph? And you could say, like, yeah, it's a smaller font, but that's just the typesetting of the journal and things like that. What do you see here? There's subsections, right? So you want to break it out into subsections that make sense. So in this case, I would recommend breaking it into subsections that, not by experiment, but by similar things. Like, for example, PCR, right? We did it twice, right? Instead of saying for the genotyping 1, genotyping 2, because that's redundant, okay? What else do you see about this materials and methods, about the structure?
A lot of simple sentences.
A lot of simple sentences don't need to be really complicated. If you're using a lot of commas and semicolons, you're way out of bounds, okay? Simple sentences. We're smart, but we're also dumb at the same time, you know? So keep it simple, all right? What else do you see? Abbreviations, right? You do see abbreviations. So if you use abbreviations, what should you do the first time you introduce that thing? You should write the full thing and then put it in parentheses after the first time. From here on out, this is what this is called. All right. What else do you see in this materials and methods section? As far as structure, let me scroll down to a different section because this is just plasmids and antibodies. Let's get to something like about right here.
It uses a lot of specific numbers.
A lot of specific numbers, but what are these specific numbers of? Concentrations. Are they initial concentrations or final concentrations? They're final concentrations.
Do the times matter?
So do the times matter? That's a great question. So there are certain techniques, I was going to get to this eventually, it's a good time to get to it now. There are certain techniques that are understood, but if you make any deviations from that technique, then you must include that specific information. So for example, right here, this particular assay, they removed the insoluble materials moved by centrifugation at 20,000 G at 4 minutes for 10 minutes. That is a significant deviation from a standard protocol of this type of assay. So they have to include that step. So think of it this way. If you're just doing a PCR, I'll just use PCR as an example. If you're doing a PCR and it's a standard PCR from the manufacturer's protocol, you say, by a manufacturer's protocol, that comes with a little handout that you get when you get the tack from the company. By the way, what we did in here was per manufacturer's protocol. Okay?
Everything? Except for the single word license.
Yeah, that was ours. I'm trying to just go back in my head and make sure. Yes, everything was by manufacturer's protocol.
Okay.
All right. But if you add or you modify them in any certain way, then you have to include information. So remember, your materials and methods, I better be able to take the materials and methods and recapitulate your experiment exactly the way that you did it. So if you made a significant deviation that could affect the outcome of that experiment, you better include that step in your materials and methods.
Okay.
Right?
I'll also say that time matters depending on your discipline. So for me, if I'm growing up a biofilm, whether it's 24, 48, or 72 hours, I would have to indicate that because those are very different stages of biofilm development, and it will matter what I'm testing or what I'm asking. So also depending on those of you who are like hours matter.
But for stuff like that we did, like 3 or 4 hours didn't really matter that much. Like if you did 3 hours or 4 hours, it wouldn't matter that much. But there's certain experiments where that variable time is essential. So you have to ask yourself for every experiment, what did I do and what were the key components of this?
For PCR, concentration of load dye doesn't matter, does it?
Well, it shouldn't because we normally, I don't know, what do y'all think about load dye? I'm usually on the fence because it's kind of assumed. Load dye and the PCR, like if you ran sample in a gel, because usually you dilute to 1x. Yeah, so I usually don't mention it. Yeah. Yeah. Because the gel won't work if you don't use load dye.
Okay.
Yeah. Yeah, so things like that. So again, a way to do it is to write it all out and then start cutting and be like, is this an important detail? Is this too much? Is there a different way I can say this, right? And then you'll start paring it down that way. Okay. Now you have gotten feedback from what materials and methods that I gave you. So use that feedback that I gave you to start writing the rest of it. Okay. So if I told you this is too creative, this is too much information, there's too many results in here, right? Then go back and remove those and try not to put any results. So your materials and methods, notice these sections should never have any results in it. You shouldn't say like, I got a band of 1,000 base pairs, let's just say, in my gel, because that's a result. Never say that. Just say I ran agarose gel. Right?
Sometimes I find myself wanting to explain why we did something. Is that something that's appropriate for like discussion? So like
Yes.
an example is like the bleach gel. Why we did the bleach gel? Okay.
So you would write that off as being like we did denaturing gel electrophoresis. So when you say denaturing agarose gel electrophoresis, let's just say, that's understood that that's what we did, right? Now when you say, in your results section, you say to assay whether we got a induction, so we ran denaturing agarose gel electrophoresis, then you could explain why the presence of the bleach in the gel should denature our RNA and give us a clear result.
Okay, that makes sense.
So you have a little more explanation there.
In the results, not the discussion.
Right? Because the discussion is the analysis, right? It doesn't matter to me that you ran a bleach gel in the analysis. It's like, what did you gain from that? Okay. So any questions about materials and methods moving forward? We've kind of already outlined our paper, so just general writing on materials and methods. Because then I'm going to group it. An A paper would group it by common technique rather than order it was done. Now, if you did it in the order the experiments were done, I think it would be okay, but you would probably lose the point because that's typically not how we write science. Would you agree?
I mean, I tell my students to write it in the order of grouping it.
Yeah.
You know, like, so in my lab, students did PCR almost every week. I don't want to know every single PCR we did, but, you know, you should mention PCR before you mention gel electrophoresis. So, like, that for me is important. Like, why would you be doing gel electrophoresis before you do PCR?
Yeah, so I would like it like that. Like, order, yes, logical order, but not necessarily by like week 1, week 2. Yeah, not by date.
Right, because the order in which you do the experiments and the way you kind of group the results together to make sense of them don't always have to be the same order in which you did the experiments themselves. Right? So you always want to make it easier to tell the story, including what were the techniques used.
So you want to like indicate like you want to indicate like what step was done for a certain.
I would.
You don't necessarily do step 1.
Yeah.
No, but I mean like in order like PCR then you would.
Yes.
Yeah. Exactly. So if you were doing those sections, you would be like, the first thing I would do is I would have a section that's PCR, boom, PCR and genotyping, and I would fill out that section. The next section that I would do, kind of giving you the answers, is agarose gel electrophoresis, and then I would talk about all the agarose gel electrophoresis things that I did, right? So on and so forth.
I'm kind of struggling to understand how we can put 2 things that happen, one at the beginning of the semester and then one at the end of the semester,
Yep.
in the same section. Like,
Yeah.
that doesn't make any logical sense to me.
Well, you can have multiple paragraphs in one section.
Yeah. So, like, would I just say we did PCR this? No.
Nope.
Yeah, I don't really understand that.
So you just have to find a way to say it. One way to say it would be to determine the genotypes of forms used in the study. We did PCR, and then break it out into to determine the genotype of the N2 versus KLP4 mutant worms. We did this with this set of primers to determine the genotype of potential knockouts later. We did this.
Okay.
Yeah. Yes.
I'm just like, building on that, like, I'm kind of confused too, because I feel like you have to, like, prep what, like, everything else that, like, happened to understand what, like, you're doing PCR of, I guess.
Yeah.
Like, I feel like you have to explain, like, the gRNA induction that you did and everything to then go on and say, like, you did PCR.
That's a good point. The thing is that you don't, because the materials and methods is a standalone section in the paper, right? So this is why we have sections in the paper, and then we have these intro results discussion materials and methods. Because if I want to know exactly what you did, I go to the materials and methods. I don't bother with the result, right? So papers, believe it or not, are not like works of fiction or nonfiction that are meant to be read page 1 to page 10 straight through, right? You should be jumping around when you read papers. A good scientist will jump around that paper and read different sections at different points. So me personally, what I do when I read a paper is I start with intro, and then I ask myself, is there something that I need to know about what they did special? Then I might go to the methods, I go back to the results, but then in the results, I'm definitely flipping back and forth between the methods, right? Even sometimes I'm going to the discussion, being like, well, how are they, once I got a result, I'm like, well, how are they interpreting this piece of data? What does it all mean? Because I interpret it differently than they do. So it's not meant to be a linear narrative, so to speak. So your materials and methods section can stand alone, right? You're just telling me the materials and the methods. You're not telling me any reason why you did anything, right? Other than if it was part of the methods, like you add an IPT to induce, but you're not telling me what was the purpose of the induction. You're saying to induce it, right? Yep. I don't necessarily think it would be wrong. I think it would be not the most correct. You would get points for it. Yes. It would be not the most correct. But you wouldn't be completely wrong. But again, there'd be some level of redundancy. And this is what I talk about when I talk about redundancy. Right? And this is why I start with materials and methods, because if you're going to have a redundant section, this will probably be the most likely to be the redundant section because we did different techniques multiple times, right? So I'm trying to give you the pointers. All right, we'll talk about another section that has a lot of redundancy in it later, but this is the big one. Okay. Any other questions on materials and methods? All right, because I'm going to move on to talk about. On to results. That's cool with everybody. All right. So I love this part of this paper. This is a really good paper. But I want you just to read the first sentence of this section and the first sentence of this section and tell me what they have in common.
To do this,
Exactly. Statement of purpose. Every section of your results section, so every subsection of your results section should start with a statement of purpose. To do this, I did this, or we did this, or to determine this, we did this. Okay? It sets the stage. You tell people what you were planning to do. So the reasoning why you did this particular experiment. To show that, or to make sure that, or to figure out this, or whatever, however you want to say it. So there should be to, like this case, to study the regulation of this, to identify, right? A statement of purpose. If I don't see that as the first sentence of each of your subsections, I question whether or not you actually understood what we were doing, the purpose of each step, right? Because it is a multi-step. So you better have that in it. I will tell you right now, that is the first thing I look for when I read your paper. Because that tells me whether or not you really understood what was going on. And then you'll notice, and I didn't have you read the rest of it, is that then they get into explaining a little bit of reason why, right? So why I did this particular thing. Like to do this, we did this, this is why we did it. And then they hit the result, okay? So I'm going to start on this one. It says, to identify the domains of KIF17 involved in auto inhibition, we created truncated versions by removing successive segments from the C terminus. Boom, right here. In text, figure call out, right? Not as seen in figure 2. If you write as seen in figure 2, I'm going to be in Washington, D.C. I will probably email you and be like, what the hell is this? Right? Yes. I might even send you like a screenshot of like me, right? Don't ever do that, okay? Always have in-text figure call-outs. Parentheses, figure 2, in this case it's figure 2C, okay? Boom, end of sentence, period. If you have a citation on the end of that sentence too, you can group those two things together in the same set of parentheses, okay? That's fine. All right?
like for our results, like we obviously have results for like different sets that we've done.
Yeah.
But like our overall like purpose was to determine like what the feeding methods that we
Yeah.
use, So should that be like should that just be like a general results section? Or
I will get to that part. Yes. Who had another question?
I just said if we did like figure one in a different citation, what would come first?
Usually the figure call out comes first. Yes. Okay. So do figure call outs like that. All right. So this one says analysis of human KIF17 sequence. Again, another figure call out. They don't necessarily need to be at the end of the sentence. It's nice if they are. It shows full range of potential. Da 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 da. Don't need to get in this. And then they kind of get in a little explanation, right? And then at the end of this paragraph, they have this sentence. KIF17, the central region, contains approximately 300 residue segment of undefined structure, which suggests that mechanisms of autoinhibition of OSM3 and KIF17 may be different. So they start to get in a little bit of explanation. Now, just to break it up, they get into another paragraph. This is actually broken into 2 paragraphs because they have one that's kind of purely structural. Because truncation of OSM3 tail domain to stabilize the dynamic state, we analyze the dynamic state, da 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 da. They say what they did. They didn't tell me they just told me the methodology that they used, right? They didn't tell me. Well, first I did this and then I did this. They said no, we used co-immunoprecipitation, so I did the method that I already talked about. That I've explained to you in the materials of method, so I don't have to get into the details of the method. I just have to say this is the methodology that I used. Right? And then they tell me what the results were, and then at the very end, they have the sentence. These results demonstrate that FL stands for full length, and truncated versions of KIF17 exist in a oligomeric state, right? They state the results, or their suggested results, right? There's no analysis, no what does it all mean. It's just the result. Well, yes. So what is the result? Not, hey, this is how I analyze this, and this is what it means. Okay? So if in our case, like, the result, this is my genotype. This is suggesting that this is the genotype, right? No analysis saying that these guys are mutants and they have locomotive defects. No, that's not the experiment that you did, right? You just did that experiment, so this is the result. Okay? So statement of purpose and state the result. So statement of purpose, a little bit of explanation, a mention of the methodology, the result. That's it. Okay? Figure callouts. Notice that you can have citations in your results section. That's okay. Typically, we don't have a lot of them, but you can have them in your results section. Right? Doctors Santiago and Brannick, do you have anything to add?
No, I just feel like whenever you're writing results and you catch yourself trying to justify or explain what it means, right?
Yeah.
Because you just say the result and that's it. The natural tendency will be to try to explain what it means and why, and then I found this. Don't do it. Wait until the discussion. Scientists are very impatient, so we sometimes like to, in the intro, you kind of give away the results at the very end. Or at least that's what we do in my business. But when you're writing results, say what it is. Don't worry. We'll find it eventually. And then, like when we ask you for your figures, the first thing you do is write your conclusion. Conclusion is an explanation of your results. That does not go in the results. The main conclusion is what synthesizing or analyzing what your results mean. So, like for my class, it's a lot of PCRs. I just want to tell me what fragments, in my case, for my students, tell me what fragments you found. And then in their conclusion, they're going to tell me what species that is based on those fragments.
In ours, we had a lot of like band, no band, size, right? So it's like it was successful or it wasn't successful, right? Or this is, suggesting that this is a mutant or this is a wild type, right? So we didn't have multiple bands. We either had band or no band, right? So results. Okay? Now, along with figures, or along with results, I want to take the time to find a nice figure. All right. So this is the bane of most of your existence, is writing the topic sentence for a figure legend. Okay. Now, I will tell you, we make it hard on you because we don't typically do multi-panel figures in this class. What makes it really it makes it difficult to get the difference between a result and a conclusion. But if you have a multi-panel figure, it makes it a lot easier to make a conclusion in that first sentence, right? So, for example, this sentence, the kinesin 2 motor kipsentine is inactive for microtubule binding in mammalian cells. So all of these figures that are above it, which I'm not going to show you all of them, lend themselves to this conclusion. Okay? So there's multiple experiments in this, multiple panels, and they all come to that, they all point to that conclusion. Alright, now again, we don't have multi-panel figures, so it's a little bit harder, but remember that first sentence must be the conclusion of that figure, what you're concluding from that figure, right? And then if you notice the text of this figure legend, they have it broken down into subsections, right? But they have all the important mention of the methodology right there. It's not an overall methodology, but it says they're untreated or treated with something called AMP-PNP, right? Fixed immunostain. They tell me what the scale bars mean. So in these images, these are all immunofluorescent images. They tell me that scale bar equals 20 micrometers, micrometers, right? So if you did a graph, and you had error bars, and then you better tell me what those error bars represent, right? Things like that. They tell me N number, at least 150 cells and 3 independent experiments were analyzed for each construct, right? So these are things that need to go in a figure legend. The figures? Okay, so this paper, since this typeset has them embedded, I want you to put all your figures at the very, very, very end of your paper. After the works cited, yes. I want the figures at the very end so it doesn't throw off the formatting. And I think the other professors want it the same way. Okay? Don't embed them in the text. I will take off points for that because the problem is because I don't know where it becomes very difficult to follow because it throws off the formatting. I don't know where your figure legend is a lot of times and your text of your paper is at times. So all the figures at the end. Okay?
This is kind of minor, but do you want them center aligned like in this one or left?
Doesn't matter. Okay, any questions on results for figures and figure legends before we go on to how would we group the results? The results, now this is where it gets different. The results, you definitely want to do them in the order that they make sense. So it would be, in our class, we did everything in order, right, for a reason, right? So you would do them in chronological, the results in chronological order. So what was the first thing that we did? The genotyping of the strains that can't be fortunate that we're mutating. Then we did the induction, right? And then we did the feeding, and then we did the sequence, right? So that is in order. Okay. You don't want to jump around. Yes, Dr. Do the order of the figures matter? What do y'all think? Yes, they matter, right? So figure 1 should go with the first thing that you did. Figure 2 should go with the next thing, right? Now, occasionally they'll jump around a little bit if you need to refer back to a figure, but they should relatively be in numerical order with the chronological order of which they should be interpreted. Yes.
I have another piece of advice since my class completed their graphs already. So one common mistake I noticed is that they all submitted figures individually, right? And we graded them, and then they just took those same figures and put them in their final paper, and they were all different font sizes, different fonts, because they just took their assignments and put them at the end of the paper. Just make sure it's all neat, same size, those things matter, and it looks kind of sloppy.
Yeah.
Don't match the paper.
Exactly. So we'll have to go back and touch some of that stuff up. Did you for which one? That's like, yeah, that doesn't go in. That was a one-off. That doesn't go in your paper. Yeah. Yeah, we don't have any graphs, but I'm just doing that for your general knowledge, right? Okay, let's. If you're getting too specific, like if you are getting way into some type of type, no, that would be results. Like as far as mentioning the methods, if you got into, well, I used these primers at this concentrate, the final concentration, and then I did 30 cycles, and I did melting, annealing, and extension. Yeah, and then we tested. You verify like that's getting too far off the end, right? Okay. Okay. We are going to move on. I'll move forward, I guess. To the intro. Okay. Now I'm going to go to the end of the intro. So first of all, we kind of talked about this briefly. As you put together your intro, what shape should your intro have? An inverted triangle, right? Starting very broad, getting more specific to our specific question. Somewhere in the end, probably the last paragraph of your intro, you should have your overall research question or overall research goal. That's where that should be. Okay? So in science, we kind of cheat. We don't like surprises, and so usually the last paragraph of the intro has, this is our overall question, and this is what we found, right? And then the rest of the paper backs up that statement. And at least in like genetics, we do that. or like my field, we do that. Yeah. Yes. Yes. Okay. So let's just look at this right here. This is that last paragraph. So it starts out, we set out to uncover the regulatory mechanisms that control the activity of BC2 motor, KIF17, both in vitro and in live cells, right? Their overall research question that paper addresses is the first sentence in that paragraph. Okay? Then, the next sentence begins with, we show that, so they spend the rest of this telling the reader what they found. Right? In the intro, the last paragraph of the intro. Okay? So this is a lot of times how we finish an intro. I'm not going to get into how we start an intro because we all have the inverted triangle idea down. But that last sentence, for sure, should have your overall research question. So when you have the inverted triangle, that point should come to your overall research question. So that should be the logical next question in that entire intro. It should be like, blah, 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 blah. We set to find out this. And this is what happened. Okay?
When it comes to like saying like weed, stuff like that, should that go in like
Yeah.
like we found really sort of carnival.
Yeah. And this is what we found.
Yeah. So like throughout the paper, like when you mention like yourself finding stuff out
Like, what do you mean? Like
yeah.
using we and I. I think you could use we and I, kind of liberally. I mean, you should try to limit personal pronouns, but you can use we and I. So what are your thoughts on that?
I feel like if you're writing now, it's appropriate.
Yeah.
Usually in results, I don't use that. It's kind of happened, right? I already know.
Yeah.
But in the discussion, perhaps, in the introduction, when you're like stating the purpose, sure.
So I would say you can use them, but just try to limit them. Don't start every sentence as we did this, and then we did this, and then we did this, and then we did this, because then that comes off as being like a list. But if it's appropriate, use it. It's used more in the discussion than usually in the results.
Can we talk about sources for a second?
Later. Sure.
Okay, because it goes in with the introduction. So that's where a lot of my sources are coming
Yep.
from. So I'll get like 3 or 4 sentences in a row that are from the exact same source.
Mhm.
How do I cite that?
How do they cite that, Dr. Brandon?
Outside of the punctuation. And then that means that every sentence before, or like in that paragraph or whatever, is from that?
Yes.
Okay, cool.
Yes.
So you don't need to have
Every sentence. If you do put it after every sentence, I'll mark it. But put it this way: I will never. If you would do it after every sentence, I would never mark off for it because then you're understanding that I need to cite that. And since we don't have a page limit on this, won't mark off for it. I'll make a comment on it and be like, "This is an easier way to do this," but I will never mark off for oversighting. Underciting, absolutely.
Oversighting is a stylistic thing. Underciting is a flagrant thing. So I tell my students, especially those who are first, learning, I'd rather you oversight than you undercite.
Yes.
Papers to campus.
Sure, this paper. Yeah, I can do that. Okay.
So that's the way learning how to write papers.
Reading papers.
So everything
Yes.
I have in this
Reading-
paragraph
yes.
is from this one.
Super style.
Yes, reading papers to get the stylistic idea down. Okay. Now I'm going to, is there any more questions about intro? I know I'm jumping around a lot. Now we're going to go into discussion. Okay. So the discussion, this particular discussion breaks out their different topics in the discussion into different subsections. You can do that if you want, but you don't have to. Okay. But notice that. So they start out their discussion with a general paragraph of the statement, right? It's kind of like a general overview of what they did, why it's important, and then a general sentence of what their overall conclusion from the paper was. Okay? Now, this particular paper, like I said, goes into different subsections. That's okay. But you'll notice that what they do is this is where the analysis and the what does it all mean, and how does this relate to what is known and what is not known. How do I fold all these ideas together, right? So I know we're not talking content too much, and I feel like I'm talking a little bit too specific about my class, but we did CRISPR. There's a lot of stuff out there about.
Everyone in here is in your class.
We did a lot about CRISPR. So there's a lot of stuff about CRISPR out there. So one common mistake I found by reading everybody's results and discussion paper is you went and you looked on like Web of Science and said CRISPR. Oh, this is an example of somebody that used CRISPR. This is another example of somebody who used CRISPR. I'm glad you found those papers. They're not appropriate, right? Just because a lot of people use CRISPR, so just because you found me 4 papers where people use CRISPR doesn't mean that they're cited appropriately, right? So every citation that you use must add to your story. It must either be evidence supporting your claims, or if it's evidence against your claims, you better cite it and counter it, okay, with something else. So I'll give you example of one of those. So let's say Smith and Jones did a similar CRISPR screen and got this many mutants, right? Suggesting that CRISPR screens are using CRISPR as effective mechanisms to do a screen, right? That's a pro. That's something that supports our position and our methodology and why we were doing what we did, right? Or I could say, but there might be a paper out there that said Thompson and Campbell attempted to do a CRISPR screen, and they found it to be a very, very ineffective methodology of doing CRISPR screen, right? Campbell and Thompson. However, they did their screen in Arabidopsis, right? We did our screen in C. elegans, suggesting that maybe it might be a nuance of the system that why theirs was ineffective, why ours might be more effective, right? So see, I did 2 things there. I did one that was pro my position, one that was against my position, right? So think of it like an argument, like you're a lawyer, like you're defending a position, your position, okay?
So do you want us to have at least one of each?
You don't need to have at least one of each. Sometimes it helps to have one of each, but you don't necessarily have to have one of each. Yeah, whatever appropriate. Yeah.
Helps you obtain what you obtain. That's better than just randomly picking papers just to say, hey, I have references here, like in my project I mentioned Horatio, but was it related to what my students did?
Yeah.
Not at all. So I was like, nope, not right.
Yeah. So I'll tell you, like, CRISPR, a lot of people use CRISPR. I can go out there and find you thousands of papers that use CRISPR, but they're not all appropriate references. Right?
I also say you may find lots of, especially with CRISPR, lots of papers that may help support your idea. It's okay if you just pick a few and not do an entire lit review of
Yes.
everything that is known about it in your discussion, right? Because then it just gets too long.
Yes.
Right? So you want to be concise.
Because we use CRISPR, should we have like a source of like what it is and then say why we use?
I mean, I'm not going to go as far as say that's required, but that might help you write an effective discussion of like what is CRISPR and why would somebody want to use it for this particular methodology that we're doing.
I was just curious how you were done it.
Well, you wouldn't want to tell me what CRISPR is per se, but you more of the
like the mechanism and
why this mechanism would help us reach our research goal, right? I don't necessarily, like that's a good point. I don't need a serial summary of what is CRISPR-Cas9 because I already know what CRISPR is.
So related to that, for the intro, then since we're starting on a broader level, then would it be unnecessary to explain, like, I guess what C. elegans is, or just why it's important?
I would stick to more of the why it's important than what it is. If you're going to say what it is, it should be one sentence maximum. Because C. elegans is a very common model organism. Yeah. Anything to add? Okay. So, any other questions on intro or discussion? Because last thing we're going to talk about, what? Well, the last two things we're going to talk about, the references. I don't need this paper for the references because this is a different journal. This is the Journal of Cell Biology, which is a pretty good journal. Make sure, some people got points taken off on the results of the discussion. Make certain, everybody, you should use Mendeley. And what cell citation, what citation styles did you use? Cell. All right. You must, however, just because you hit insert bibliography does not mean that you are done. Right? You must go back and make sure Mendeley formatted your references the way that you intended them to be. No, I don't want it in columns like this. I want it, regular. Right. Yes. I'm looking for that it's formatted like cell, and I will give you all a hint about how I know that it's in cell format or not. If there's something called a DOI number that ends your citation, it is in the wrong format. So the last thing that should be in a cell format is the page numbers of that edition or volume of the journal. So I can look at it and be like, no, that's the wrong format. Right? So sometimes, not all the time, but sometimes Mendeley will screw up the way that it formats your citation. It'll drop a required component, like the journal title or something. So make sure you go back and you check and make sure Mendeley formatted your citations correctly. How many citations, primary sources, must you have for this paper?
10.
My class must have 10. Yes.
Another thing, Mendeley doesn't really format scientific names correctly. So for me, bacterial names have to be italicized, and if there's bacterial name in the reference, it will not be italicized, so I have to go back and do that manually. Same thing for C. elegans.
C. elegans must be italicized. It is a proper Latin species name. I didn't take off for it before, but I will take off on it. Yes.
One of the things I noticed with Mendeley is that it'll abbreviate journal titles.
Yep.
Is that incorrect?
No, that's correct. It abbreviates, if you tell it cell, it abbreviated for the accepted abbreviation of that journal. Okay. Now we're getting into some of the formatting things. So, how should your works cited section be formatted? It's in cell format, but how should those entries be formatted? Well, it should put them in numeric. It should put them in the order that cell does it. Well, how should they look on the page within that format?
Justified.
Single space within each citation and one blank row in between your citations. Should there be any indentations of these citations? No. So your paper itself should be double spaced. I like my citations single spaced, and I like my figure legends single spaced. And I like my papers fully justified. I don't like the jagged edge on the ring. I want it straight. I want to look at.
On floral smelling paper.
No, I'm put it in. Yeah. Yes.
You want the margins to be normal.
Yeah, margins be 1 inch. All right. So that's how it should be formatted. So go back and check the formatting. So the last thing that you should do before you submit your paper is to make sure all the formatting is correct. Okay. A couple more things. What's the last thing that you should write for your paper?
Abstract.
Your abstract. You're required to do an abstract for this paper. Yes. Okay, so once you've tied your paper together, write an abstract. So abstract should be a paragraph summary of everything. Okay? That should be the first thing in your paper, chronologically, the last thing you write. All right, and your paper, make sure your paper has a title. Right? Short title. It's short, but yet descriptive.
Creative. Fluffy.
Usually fluff. Yeah, well, yeah.
The GRA word.
Make it a scientific. Okay, So I'm just finishing up here. Last couple things. So remember, you need to submit this paper. So I'll put it this way: a week from today, next Tuesday, bring whatever you have, draft whatever you have. We're going to do peer review next Tuesday in lab. Okay. So bring. If it's just bullet points, bring your bullet points. I hope you have more than that, but bring that, whatever you have. Okay.
Do you want a title page or just have the title?
I don't really care about title page because it's like a waste of paper to me. To have like just the title and your name on one page. Yeah.
So, introduction, that last paragraph, sort of like purpose and what you did.
Yeah.
So we differentiate that from the abstract.
Well, the abstract will also have that kind of information in it as well.
So it's kind of be include a little bit of the methods as well. So
Yeah.
some of your heavy piece and some of that.
So it's overall summary. Yes.
It's
In one paragraph. Yeah, it's weird, but this is how it's done.
Another like minor formatting thing. So between like the different sections, like the abstract, the intro, etc., that should we have page breaks between them?
You can just do continuous as long as you have section headings. Okay. Sebastian or Ben, you have questions?
With the original figure, I know this is a lot that it can kind of like summarize like the whole
Yep.
experiment. You know, is there a way to do that instead of just like visualizing the steps? Like how?
I leave that up to you. Be creative. I leave it up to you.
Don't forget to cite your original figures.
Yeah.
Put all of this work and time and effort into it. Write the paper and don't cite it once. Bad idea.
Yes. Okay. So yeah. So next Tuesday, bring whatever you have, whatever kind of draft you have, to lab. We're going to do peer review. On December 6th, Friday, December 6th, your paper is due to 2 different places by 11:59 p.m. What are those 2 different places? Oleotech and Canvas. Okay. I will have a Turnitin link on Canvas for you to upload your paper to, and I will have the Oleotech link should already be on there. Okay. Yeah. Okay. Last thing I want to do before. So I just want to show you something. So this is, I just want to show you how writing. That's my daughter. Okay. I just want to show you how writing is an iterative process. So my wife and I are actually writing this paper together right now. You know, it doesn't matter what it's about. It's about CRISPR. But this is what?
That's cool.
Yeah. But writing is a very iterative process. Like this, it says like version 3 of this paper. This is pi version 3. This is probably like the 6th or 7th version of this paper overall, right? Now, I know that we're running short on time, but it's always a work in progress. So always keep versions of it. Always be tweaking it. Always be sure. Because remember, it's important. It's the currency at which scientists have is your publication record, what you've done. I mean, as scientists, they want us to be excellent teachers, and we all want to be excellent teachers, but we're also scientists, and we're judged in our respective fields by the research that we do, and the best way to judge our research is by the papers that we publish, okay? And so it is an iterative process, so it should have markups on it. You should go through and you should be like, I don't like how this sounds, I need to reword it. Is this actually what it means? Is this what I actually intended to say? I have an idea of what you want to say. I know what we did. The best way to prove to me that you know what we did is make it very, very clear in your writing. Okay? So even though you write a sentence and you're like, well, I have a topic sentence in my results, but I'm like, read that sentence. Is that actually what we did? Right? Because I found a number of people on their results and discussion that they had a statement of purpose like 2 do X, we did Y, but it was something like way different than what we did. And then when I read that, I'm like, okay, did they really understand what they did? If I can understand that you understand what we did, that's an A paper, right? But if it's really kind of convoluted, and it's like run-on sentences, lots of semicolons and commas and all this other stuff, Then I'm like, what? Did they really get what's going on? Okay? So make sure your writing is clear and it's an iterative process. So if you get a peer review next week and somebody just rips it apart, that's a good thing, right? That's getting feedback before you hand in the real one.
So I'll tell you a couple of things. So one thing is, I'm a perfectionist. Okay, just get words on the paper. I spent so much time during graduate school trying to make my sentences perfect, when all I needed to do was word vomit on the page and then go back and make it perfect. And if you're a perfectionist like me, you are always going to find something wrong with it. So you have to come to a time where you're like, this isn't good enough, okay? And I just need to be okay with this because you're never going to be happy with it otherwise. Okay, so just get something on the page and then you can go back and smooth it out. Another thing I will say, read it out loud. It makes sense to you because you, like, especially if you're working on it, like Dr. Mike says over and over again, you know what you want it to say, and it starts, as you read it, not aloud, makes sense to you. But when you start reading aloud, you're like, that does not even make anywhere close to sense. Okay, so I will read. People probably come in near my office and probably think I'm talking to myself. It's because I'm reading papers that I'm writing out loud to myself.
So to piggyback off that, I know we're running short of time, but if you are under the impression that you are going to be able to write this and write it all the way through from first word to last word, hit format bibliography and be done and hand it in to me, that's not going to be enough. I will tell you right now, even the best writers of you, that's probably going to be a C paper. That's the way it works, right? So over the next couple weeks, work on it. It doesn't have to be all done in one shot. A little bit here, a little bit there. Compile what you have. Figure out how to work on it.
And TJ, biology tutors.
Yeah.
Writing and science is a lot different.
All right. Any other questions?
Read the best draft possible.
Yes.
Put on paper, work on the larger get feedback on.
Say goodbye. Put a little Easter egg in it.
Dr. Santiago, I need you to log into your computer.
Okay, so like, can I ask a question about our experiment?
Yes. Well, can I end this
Oh.
this movie that was almost an hour, like 5 gigabytes?
Oh, I'm in it.
Yay. Later.